In this video, we are going to look at question 3 for exercise on chapter photosynthesis. Okay, we are going to look at chapter 3 and also question 4. Okay, question 3 and 4. Okay, so for question number 3, A, figure 5 shows a simplified Kevin cycle of photosynthesis. So as you know, Kevin cycle already mentioned in the previous video that it has 3 stages. Okay, the first stage is carbon fixation. Carbon dioxide is incorporated with carbon dioxide receptor, which is RUBP, in the carbon fixation stage, the first stage, to form the first product of carbon fixation, which is a 3-carbon compound, which is we call it as PGA, or 3-phosphoglycerate. Okay, 3-phosphoglycerate. So PGA is basically a 3-carbon compound. Okay, because we are focusing in this case, we want to know the production of G3P for 1 molecule of glucose. So in order to produce 1 molecule of glucose, it requires 2 molecules of G3P. Okay, 2 molecules of G3P. So because we want 2 molecules of G3P, so the cycle will consume 6 molecules of carbon dioxide. Okay, so from the 6 molecule of carbon dioxide, it combines with 6 molecule of RUBP to produce a 6 molecule of 6-carbon compound. Okay, 6 molecule of 6-carbon compound here. So if I were to draw, okay, so this is the carbon from carbon dioxide combined with carbon from RUBP. RUBP has 5 carbons, so 1, 2, 3, 4, 5. So the molecule has 6 carbon. Okay, 1, 2, 3, 4, 5, 6. So this molecule is not stable, so it's split into 2. Okay, 1 and 2. Okay, so because initially we have 6 molecule, so when it breaks into 2, so 6 times 2, you produces 12 molecule of 3-carbon compound. Okay, so this carbon compound produced, we call it as PGA. So 12 molecule of PGA, or 12 molecule of 3-phosphoglycerate. Okay, so this occurs in the first stage, which is carbon fixation. So then it enters into the second stage, which is reduction. Okay, so because 12 molecule is produced in the first stage, 12 molecule of PGA, so in the second stage, it will consume 12 molecule of ATP, okay, to produce 12 molecule of 1,3-bisphosphoglycerate that will be reduced by NADPH. So how many molecule of NADPH will be used? 12 molecule also. Okay, so this 12 molecule of NADPH will reduce 1,3-bisphosphoglycerate into 12 molecule of G3P. Okay, so G3P again is a 3-carbon sugar. So from this 12 molecule of G3P, 2 will exit the cycle. Okay, 2 G3P will exit the cycle to combine and produce 1 molecule of glucose. Okay, from 12 minus 2, 10 will proceed into the 10 molecule of G3P will proceed into the regeneration stage. Okay, regeneration of what? Of RUBP, which is carbon dioxide acceptor. Okay, so in order to rearrange, okay, the carbon in 10 molecule of G3P, the purpose of the third stage here is to produce 6 molecule of RUBP again. So in order to produce 6 molecule of RUBP, so therefore 6 molecule of ATP will be used during the third step. Okay, to rearrange 10 molecule of G3P to form 6 molecule of RUBP, to regenerate back the carbon dioxide receptor to repeat the cycle again. Okay, so altogether, okay, to form 1 molecule of glucose, you have to know that the process consumes 18 ATP molecule and also 12 NADPH molecule. Okay, it also consumes 6 molecule of carbon dioxide. Okay, so name molecule A. Molecule A is carbon dioxide. Okay, so which stage of photosynthesis produces NADPH? NADPH and also ATP? The answer will be during light reaction. So light reaction, as you know, the purpose of it is to produce these two molecules, ATP and NADPH, that will be used during the Calvin cycle. Okay, so next question, show how many ATP is consumed if a plant needs to synthesize 3 molecule of glucose. Okay, tadi kita dah tahu kan, untuk 1 molecule glucose dia perlukan 2 molecule of G3P. So untuk menghasilkan 2 molecule G3P ni, okay, dia perlukan berapa? 18 kan? 18 ATP, 12 NADPH, 6 carbon dioxide. Soalan ni dia tanya untuk 3 glukos kan? So, berapa banyak ATP yang diperlukan? So, awak dah tahu kan tadi, 1 glukos dia perlukan 18 ATP. So, okay, so maksudnya 2 molekul G3P, nak menghasilkan 2 molekul G3P, the Calvin cycle will use 18 ATP molecule to produce 1 glucose. So in order to produce 3 glucose, okay, so 18 ATP, 18 ATP times by 3 molecule of glucose going to be synthesized. So the answer will be 18 times 3, you get 54 ATP will be used during Calvin cycle to produce 3 molecule of glucose. Okay, so figure 6, next question. Figure 6 shows part of photosynthesis in a maize. Maize is corn plant. Okay. So as you can see here, basically it shows you light reaction. To be specific, it shows you the process of linear electron flow, okay, or non-cyclic electron flow from here, okay, from photosystem 2 to the ETC, to photosystem 1 to ETC, and then received by NADP plus here, okay, to form NADPH. Okay, it also gives you the flow of electron if the electron flows in a cyclic flow, okay, cyclic photophosphorylation, which is from photosystem 1, okay, to primary electron acceptor, to ferredoxin, to cytochrome complex, to plastocyanin, and back to photosystem 1. So ada dua lah, linear electron flow which produces both ATP and NADPH, linear kan, daripada photosystem 2 sampai elektron tu digunakan untuk menghasilkan NADPH. So linear electron flow will produce both ATP and also NADPH. Kalau untuk cyclic electron flow or cyclic photophosphorylation, it only produces ATP molecule through the process of chemiosmosis. Okay, so now we are going to look at the question. Okay, so question 1, state the specific location of X in the plant organelle. So plant organelle involving here is obviously chloroplast. Specific location in the chloroplast is the thylakoid membrane. Okay, so thylakoid membrane, as I've mentioned, it is the place where light reaction occurs. So in order for light reaction to occur, all the photosynthetic pigment, photosystem 2, and also electron carriers must be embedded in the thylakoid membrane. Next question, explain the significance of Y. So Y is NADP plus reductase, okay, which is an enzyme used to receive electron from ferredoxin and then transferred to NADP plus to form NADPH. Okay, so the question is 2 mark. So basically Y, if you mention Y is NADP plus reductase, you get 1 mark. And the function of the enzyme is to catalyze the formation of NADPH or reduces NADP plus to NADPH. Next question, explain how energy released from electron transport is used to form ATP. Okay, so let's delete all this scribble. Okay. Okay. Alright. So you know that, okay, electron from photosystem 2, to be specific from P680, okay, will be excited, okay, through the absorption of photons by the accessory pigment and received by P680. So the electron of P680 will be excited and then received by the primary electron acceptor. Okay, so this electron will have, okay, so then the electron received by the primary electron acceptor will have high energy, okay, as the result of absorbing photons, okay, photons and release. The photon will excite the P680 and release the excited electrons. So the electron will be transferred along ETC, okay, along the electron transport chain from plastoquinone to cytochrome complex to PC. So bila tengok gambar ni, dia makin menurun. Sebab apa? Electron yang travel from one carrier to another carrier will release energy. Okay, so this energy is basically used to synthesize ATP using chemiosmosis. Basically, the electron, okay, the electron will be used to pump hydrogen ion, okay, to pump hydrogen ion from stroma into thylakoid space. So kalau saya draw kat sini untuk fahamkan proses dia, okay, ini adalah chloroplast. This is the chloroplast that has double membrane, okay. The fluid inside the chloroplast, we call it as stroma, okay. The concentration of hydrogen ion in the stroma has to be low. Okay, inside you have the thylakoids, okay. The thylakoid space, okay, the thylakoid space, it has to have a high concentration of hydrogen ion. Okay, so in order to concentrate the hydrogen ion into the thylakoid space, alright, the hydrogen ion in the stroma has to be pumped, okay, into the thylakoid space. So the pumping, the source of energy to pump the hydrogen ion from stroma into the thylakoid space, the source of energy comes from the transfer of electron. Okay, so the transfer of electron will release energy to pump hydrogen ion from stroma into thylakoid space, so that in the thylakoid space it has a high concentration of hydrogen ion. Okay, so that's why, okay, the transfer of electron releases energy. Okay, releases energy. So the purpose of pumping hydrogen ion into the stroma, the purpose of pumping hydrogen ion from stroma into the thylakoid space, so that when you have a high concentration of hydrogen ion in the thylakoid space, it creates proton gradient. Okay, proton gradient or proton motive force. So this proton motive force will be used by ATP synthase to synthesize ATP molecule during chemiosmosis. Okay, so the question asks you, explain how energy released from electron transport chain is used to form ATP. Okay, the question is worth 3 marks. So let's look at the answers. Okay, as I said before, movement of electron releases energy. Okay, so this energy is used to pump hydrogen ion from stroma into thylakoid space. So the purpose is to create proton gradient or proton motive force, or to make sure inside the thylakoid space it has a high concentration of hydrogen ion. So this is important so that this proton motive force or the high concentration of hydrogen ion can be used during chemiosmosis, okay, which is the proton motive force will be used by ATP synthase, okay, synthase to synthesize ATP from the movement, okay, from the movement of hydrogen ion that diffuses, okay, from the thylakoid space back into the stroma through the ATP synthase. So basically the purpose of generating proton motif force is that it is a source of energy used by ATP synthase, okay, to synthesize ATP. So when you have a high concentration of hydrogen ion in the thylakoid space, the hydrogen ion will diffuse back through the stroma using ATP synthase, and the ATP synthase will use the proton motif force to synthesize ATP molecule from ADP and also inorganic phosphate. Okay. So hydrogen ion diffuses from thylakoid space back to stroma to ATP synthase, and the ATP synthase synthesizes ATP. So any of this point, okay, you choose 3, you will get 3 marks. Okay, so basically you have to understand the whole process. Okay, the whole process of why electron needs to be transferred from one carrier to another carrier, and how it relates with chemiosmosis, and lastly in the production of ATP molecule. Okay, so ATP basically, you know, it is required during Calvin cycle to produce sugar. Okay, so for the next question, question C, figure 7 shows absorption spectra of plant pigments. Okay, photosynthetic pigment absorption spectra. It shows you a graph that shows the wavelength of light, okay, absorbed by each pigment. Okay, pigment Z, pigment Y, and pigment pigment X, Y, and Z. Okay, absorption spectra is a graph to show you the amount of light energy absorbed by individual pigments. Okay, so what is absorption spectrum of photosynthesis? So basically question 1 asks you for definition. So the mark for definition is usually 1 mark. Okay, so it is a graph showing the wavelength, okay, wavelength of visible light absorbed by each pigment. Okay, that is the definition for absorption spectra. Yes, absorption spectra or absorption spectrum. Okay, so number 2, describe the absorption spectrum for X. Spectrum is singular, dia untuk menggambarkan satu pigment. Tapi kalau spectra ada banyak lah kan, so that's why dia use kat sini absorption spectra, ada 3 pigment X, Y, and Z. Okay, so ada 3 pigment, so dia plural, so absorption spectra. Tapi kalau satu, absorption spectrum. Okay. Okay, so question 2, describe the absorption spectrum for X. So X, as you can see, it absorbs light. Maximum is around 400, okay, to 500 before 500 here. Okay, so roughly lah the range of that. So you know that this range is in the range of violet blue. Okay, violet blue, 400 to 500 nanometer. Okay, so describe the absorption spectrum of X. So 2 marks. Absorb violet blue light. Okay, most light absorbed in the range of 400 to 500 nanometer. So please write the unit. Or less absorption if more than 500. Okay, so 500 and 650 nanometer, the pigment will absorb less. Okay, and as you know, okay, when you exceed the 500 wavelength, so it is in the range of green light. So the green light will not be absorbed, but the green light will be reflected and transmitted, okay, by pigment X. Okay. So basically, pigment X is basically, it gives you the absorption spectrum of chlorophyll A actually. Okay. So suggest the effects if Y and X are absent in plant. Y and Z are absent, tak ada. So Y and Z, because X is chlorophyll A. Chlorophyll A is the main photosynthetic pigment. They are found in the reaction center of photosystem 1 and photosystem 2. Y and Z are accessory pigment. Okay, it can be chlorophyll B and also carotenoids. Okay, For example, chlorophyll B and carotenoids. So if the accessory pigments are absent, so it means that the rate of photosynthesis will be reduced. Why does the rate of photosynthesis will be reduced? Because when the accessory pigments are absent, okay, the other range of light, especially for example in the range of green light, which is absorbed by the carotenoids, will not be absorbed. Okay, so this excess energy that's supposed to be absorbed by the accessory pigment is absent. Okay, when the excess energy, the excess light energy is not absorbed by the missing chlorophyll, so therefore the rate of photosynthesis will be reduced. Itu fungsinya accessory pigment ni, untuk memastikan lebih banyak sumber cahaya matahari boleh digunakan, boleh diabsorb untuk memaksimumkan rate of photosynthesis. Bila tak ada, okay, bila tak ada maksudnya hanya chlorophyll A je yang present. So chlorophyll A boleh absorb at that certain range of wavelength of light sahajalah. Tak ada excess source of energy absorbed by the accessory pigment to increase the rate of photosynthesis. So if absent, so therefore rate of photosynthesis will be reduced. Okay, so when rate of photosynthesis reduced, so therefore less glucose or less photosynthetic product or yield will be produced. Okay, less glucose produced because less light energy will be absorbed. Okay, so the next question, the last question, which is question 4. Question A, give one example of photoautotrophs other than plants. Okay, so ini ada dalam bahagian depan nota lecture note awak. So they can be cyanobacteria. So these are bacteria that are capable of carrying out photosynthesis. Same goes to purple sulfur bacteria and also algae. Okay, so any one of these organism gets 1 mark. Question B, figure 8 shows a photosystem located in the thylakoid membrane. Okay, so as you can see, so this is the photosystem. It can be photosystem 1 or 2. Okay, so K is the reaction center where the special pair of chlorophyll A will be located. Kalau photosystem 2, it is P680. Kalau photosystem 1 is P700. L is primary electron acceptor. Okay, so identify K. K is a special pair of chlorophyll A in the reaction center. L is primary electron acceptor. Describe photolysis of water in light reaction. So this one we have discussed previously in the previous question. So photolysis of water, basically it involves splitting of water molecule. Okay, splitting. The keyword is splitting. Okay, splitting of water molecule. So split into what? Into two electrons. Okay, 2 hydrogen ions and 1 oxygen atom. Or another form that you can write. Okay, so this is 1 mark. This is 1 mark. Another form you can write is 2 molecule of water split into 4 hydrogen ion, 4 electrons, and also 1 molecule of oxygen. Or in another form, 1 molecule of water split into 2 hydrogen ion. Okay, 2 hydrogen ion, 2 electrons. Okay, and half oxygen molecule. Okay, so ini semua ni cara yang berlainan awak boleh tulis. Okay, split into apa? Okay, so 2 markah. So point yang lain yang another point that you can include is that the process of splitting of water molecule requires or catalyzed by enzyme. Okay, so question C, figure 9, okay, shows a simplified diagram of Calvin cycle. Okay, again, question on Calvin cycle. So we see so many Calvin cycle question occurring several times. So please pay attention to Calvin cycle, I guess. Okay, you have to understand Calvin cycle really well. Okay, so number 1, identify molecule M. So molecule M, is RUBP, ribulose biphosphate, which is carbon dioxide acceptor. Question 2. Describe the event that produced G3P, glyceraldehyde 3-phosphate, in phase 2. Phase 2 is what? Reduction phase. Okay, reduction phase. So, okay. So during first phase, you know it produces PGA. Okay, or 3-PGA or 3-phosphoglycerate. So during phase 2, this molecule 3-phosphoglycerate, which is the first product of carbon fixation, okay, the PGA molecule, okay, kita boleh tulis dia sebagai PGA juga, right? 3-phosphoglycerate receives a phosphate group from ATP. So maksudnya semasa phase yang kedua ni kan, semasa phase yang kedua ni dia akan menerima ATP kan. ATP will involve in the reaction, and it also receive NADPH in the reaction during phase 2. Okay, during phase 2. Because soalan ni dia menggunakan 3 carbon dioxide. Okay, so maksudnya kat sini akan menggunakan 6 ATP dengan 6 NADPH. Tengok balik kenapa 6? Faham kan daripada previous video tu. Previous video yang saya dah buat. Okay, so alright, so the PGA molecule will receive phosphate from ATP molecule. Okay, ATP. So the PGA will be converted into a molecule, we call it as 1,3-bisphosphoglycerate. Okay, 1,3-bisphosphoglycerate, and because NADPH enters into the reaction, so this molecule 1,3-bisphosphoglycerate will be reduced by NADPH to produce G3P. Okay, to produce G3P, glyceraldehyde 3-phosphate. Question 3. Explain what happened if phase 3 of Kevin cycle does not occur. So what is phase 3? Regeneration of RUBP. If regeneration of RUBP does not occur, so RUBP cannot be produced. Okay, RUBP cannot be produced. So when RUBP cannot be produced, so therefore the amount of G3P will become less. Okay, will become less because the G3P will be used up. What does it mean by will be used up? Because as you see, okay, as you see here, the some of the G3P will exit from the Kevin cycle and will be used to synthesize glucose. Okay, so if the G3P is not recycled, lama-lama G3P akan berkurangan. Okay, alright. So question 4 is how many ATP and NADPH are required to make one glucose molecule? Okay, so to make one glucose molecule, it requires 18 ATP molecule and 12 NADPH. Please listen to my first video to understand why 18 ATP and why 12 ATP are required to produce one molecule of glucose. I have already explained this in the previous video. Okay. So question D, alright, the last question. If a pineapple plant, okay, is grown in an uncontrolled environment where it is provided with a constant amount of light, show how would this continuous exposure to light affect the plant? Okay, pineapple, you have to know it is a chem plant. Okay, so chem plant will undergo carbon fixation twice. Okay, carbon fixation twice. One during the night time, second during daytime. Okay, so during night time, the first carbon fixation occurs so that the first carbon fixation occurs because during night time, stomata open, carbon dioxide can enter into the leaf, and the carbon dioxide can be fixed. okay, during first carbon fixation, and produces oxaloacetate. Okay, so and then oxaloacetate converted into malate, and malate during daytime will be split into carbon dioxide and also pyruvate. So the carbon dioxide produced during daytime is crucial, okay, to be used during Kelvin cycle. Okay, during Kelvin cycle, which is the purpose of Kelvin cycle is to produce sugar, glucose. So if the pineapple plant, okay, which is a chem plant, is constantly exposed to light, so it means that the pineapple plant is continuously in the daylight condition, okay, in the daylight condition. So that's mean the pineapple is not experiencing nighttime. Okay, so when the pineapple plant does not experience nighttime, the first carbon fixation cannot occur, and carbon dioxide cannot be accumulated in the leaf of the pineapple during daytime. So the concentration of carbon dioxide in the leaf during the daytime will be reduced, and it cannot fit into the Kelvin cycle, and sugar cannot be produced. So pineapple ni mesti kena ada waktu malam untuk dia melakukan first carbon fixation tu, untuk fit carbon dioxide pada waktu siang semasa Kelvin cycle untuk menghasilkan sugar tu. Okay, so kalau tumbuhan ni sentiasa dipancarkan dengan cahaya, okay, so dia stomata pineapple tu will not be open. Okay, stomata of the pineapple leaf will not be open, and so therefore no carbon fixation can occur. When no carbon fixation can occur, carbon dioxide in the leaf will remain low, and no Kelvin cycle can occur. Okay, so when no Kelvin cycle occur, no sugar will be produced. Okay, if you answer just okay, the pineapple die, your answer will not be accepted. So you have to explain why, right? So that's the end of question 4, and that's the end of discussing chapter exercise on chapter photosynthesis. So I hope you understand what have been discussed. So please do your revision, understand the question, understand the process of photosynthesis. Okay, so thank you.